Organic is a word that is used to mean many things. Food manufacturers use it to mean vegetables that have not been sprayed with insecticide. Beauty companies use it to mean products that have been made from all natural ingredients. But what is really organic? From a biological point of view, organic molecules is a term that refers to any molecule that contains carbon. Carbon is used in many living organisms because of its atomic structure and the way in which it allows for molecular diversity in life. To see why, we must take a closer look at carbon. As an element, carbon has a medium electronegativity level. This is important when it forms intermolecular bonds with other atoms, since it allows it to form covalent bonds. If it had strong or weak electronegativity, it would increase the chance of it forming ionic bonds, and since our bodies consist mainly of water, the polar water would quickly dissolve these ionic bonds. If we look at the carbon atom, we see another property of carbon that is important, and the main reason why carbon is perhaps the most crucial element when we consider living organisms. There are 4 valence electrons in the shell, and each can form a covalent bond, meaning that the electron has a valence of 4, the maximum valence. This accounts for the diversity of life, since carbon can be arranged in a variety of ways with many different atoms. This property of carbon allows big molecules to be formed, called macromolecules. Organic molecules as a single unit are known as monomers, and specific classes of monomers can join together to form polymers. One example of this is glucose, a monomer consisting of carbon, hydrogen, and oxygen. Molecules of glucose are known as monosaccharides and join together to create polysaccharides, or carbohydrates. In this case, glucose would be the monomer, and the polysaccharide it joins to create would be the polymer. But how are these polymers formed? Polymers are formed by monomers via dehydration synthesis, or condensation reactions. This process occurs from the removal of 2 atoms of hydrogen and 1 atom of oxygen, or the removal of a water molecule. As the hydroxyl group of one monomer and the hydrogen of another monomer is lost, the 2 monomers form a hydrogen bond. When polysaccharides need to be broken up, cells do this via hydrolysis. Here, bonds between the monomers are broken up by adding water to the polysaccharide. The hydroxyl group and the hydrogen ion attach to each monomer, and the bond between them breaks. There are 4 important classes of organic macromolecules: carbohydrates, proteins, lipids, and nucleic acids. In all these molecules, the structure-function relationship is clearly visible. Carbohydrates are polymers of glucose molecules, and the structure of these glucose molecules can vary. As can be predicted from the structure-function relationship of molecules, the variation in structure causes the function of the finished polymer to also vary. In alpha glucose molecules, the OH of the first carbon points down, and in this case they bond with other alpha molecules via dehydration synthesis, in the bond form called alpha linkages. Alpha linkages cause the finished polysaccharide to be in the structure of a helical compact structure. The function of polysaccharides synthesized from alpha glucose is energy storage, and so this relatively compact structure allows more energy to be stored in a given space. An example of such a polysaccharide is starch, which plants use to store energy. In beta-glucose molecules, the OH of the first carbon points down. This small structural variation